Hey biologists, this is part 2 of your responding to antigens drawing that we did in class the other day. So let's have a look together at where we've got to. So this virus has overcome the first line of defense, the barriers, and the second line of defense has kicked in, and some phagocytes, we've got inflammation, some phagocytes have engulfed the virus, and the two that we have here, our dendritic cell and macrophage, have digested it and are presenting fragments of that virus, so the antigens, on their MHC2 markers. And they are travelling around the lymphatic system to the lymph node, where they are most likely to encounter B and T lymphocytes, which are the cells involved in the adaptive immune response. So some things that you need to know about B and T lymphocytes. They have a lot in common. So they both originate, at the bottom here you've drawn the bone marrow. They're both produced in the bone marrow, all white blood cells are. B lymphocytes are matured in the bone marrow, whereas T lymphocytes are matured in the thymus. So it's a good way to remember it. T standing for thymus and B standing for bone marrow. So that's the big difference between those two, and it's a classic question that you might be asked. Both of those types of lymphocytes are found in very high concentrations in the lymph nodes, and it's really important to know that of the millions and millions of cells that are there, they all have unique receptors on their cell membrane, and they are derived from random genetic changes during the production of these cells. Just get rid of that. So they are all completely different to each other, and by pure coincidence, they will match a particular antigen. So when they are sitting in the lymph node, but they haven't yet met an antigen that matches their specific receptor, we call them naive. So they are mature, they've matured in their specific sites, they are sitting in the lymph nodes, but they haven't been presented with an antigen that matches their receptor, so they haven't been activated, and that's what we call a naive lymphocyte. So what I'd like you to know is that the B lymphocytes are involved in something called humoral immunity. So the humoral immunity only targets the pathogen, circulating pathogen, so free pathogen, not inside your body cells, and aims to eliminate that pathogen by producing lots and lots of antibodies that again is specific to that pathogen and has a specific variable region on the antibody. If you look down here at the diagram you drew, that will match the pathogen and immobilize it, agglutinate it, so stick it together, prevent the spread of the pathogen and prevent the pathogen from infecting body cells. And then the complement system will activate, and those pathogens will be engulfed by the innate immune cells and by phagocytes. So that's the role of the humoral immunity, and that's what the B lymphocytes aim to do. Now, how do we activate a naive B lymphocyte and take it from the point of it being naive and sitting and waiting for that antigen in the lymph node to a point where it can differentiate and become a plasma B cell, or alternatively, a naive B lymphocyte can differentiate into a memory B cell, and it has antibodies, bound antibodies on its outside, and sits and waits for another infection later down, maybe many years down the track, with exactly the same pathogen, so that the response will be much, much quicker the next time around. So how is that B lymphocyte going to be activated? What could happen is the membrane-bound antibodies on the outside of a naive B lymphocyte, they could bind to a specific pathogen, and what we do know is that B lymphocytes are capable of engulfing pathogens. So in this case I've shown the virus actually has been engulfed by the B lymphocyte, and B lymphocytes are also antigen presenting cells. So what they can do is present the antigen from that pathogen on their MHC2 markers, and they can actually present that to a helper T cell. Once they've been partially activated, a B lymphocyte will begin to differentiate, so become those two types of effector cells, the memory cells and the B plasma cells. They usually need a little bit of extra help in finalizing that activation process, and that extra help comes from the helper T cell, which is a naive helper T cell, can be activated also by presentation of an antigen on an MHC2 marker. Now you'll know that these two types of phagocytes, your dendritic cell and your macrophages, have MHC2 markers, and they can present an antigen to a helper T cell, but the B lymphocyte has an MHC2 marker, and it can also activate a helper T cell by presenting the antigen on its MHC2 marker, and that's almost like a protective mechanism. It's a bit like a double handshake. So not only has the B lymphocyte encountered the pathogen, but it also then matches its pathogen, presenting on the MHC2 marker, to a helper T cell with exactly the same receptor that matches that antigen. Now if that happens, you get this chemicals being released from the helper T cell, activation of that helper T cell. So that will become effector helper T cells that will release cytokines and coordinate the attack or the action of multiple different immune cells, including some in the innate immune response and some in the adaptive immune response. In addition, the naive helper T cell will also become memory helper T cells. So again, waiting for another presentation of that antigen later on, many years later, so that it's more likely to activate the adaptive immune response. And these are things that will become important when we look at immunity and vaccinations, and why that second exposure is much faster and much larger. You'll be able to relate it to the action of lymphocytes and these memory cells that have been produced as part of this first exposure. So far what we've got is full activation of these B lymphocytes, both by it engulfing the pathogen and by presentation of the antigen to the T helper cell, the naive T helper cell, which in turn activates and releases cytokines which can finalize activation of the B lymphocytes. And then once we've created these effector cells, these plasma B cells, they will start pumping out huge volumes of antibodies to trap, stick together, immobilize the pathogen and prevent it from infecting our body cells. Let's have a little look at another kind of T cell. So we also have the cytotoxic T cells, which exist in the lymph nodes in the naive state as well. The helper T cell and the cytotoxic T cell have slightly different receptors and are activated in slightly different ways. Knowing that the cytotoxic T cell its role in the immune system is to eliminate infected body cells, that's actually activated by presentation of an antigen on an MHC1 marker. So an infected body cell can activate a naive cytotoxic T cell. And the purpose of that is so that naive cytotoxic T cells can quickly identify infected body cells or cancerous body cells and become like the very specific assassins in the immune system and induce that body cell to actually undergo apoptosis and kill itself. So these naive cytotoxic T cells are also waiting in the lymph nodes, and they also have exactly the right receptor for the antigen that's infecting the body, that's presented on the virus and is infecting the body at that time. Now, what will happen is a macrophage, for example, will present evidence or an antigen for that specific pathogen on its MHC1 marker. Okay, so remembering that it's taken it in and digested that virus or that pathogen and has MHC2 markers to activate the T helper cells, but also is presenting that antigen on an MHC1 marker, which is capable of activating these cytotoxic T cells, which have what we call CD8 positive receptors on them. So these two types of naive T cells are activated in different ways. Might not be something you're assessed on, but is something that's useful to know. Once these cytotoxic T cells have been activated, they become again memory cells and effector cells. And effector cytotoxic T cells are responsible for eliminating infected body cells either by the death ligand pathway of apoptosis or by releasing perforins that induce cell death in slightly different ways, so damaging cell membranes and damaging the inside of the cell. So as I said, these T lymphocytes are involved in the humoral cell-mediated response, and the B lymphocytes are involved in the humoral response. Humoral eliminating the pathogen from circulating, and cell-mediated eliminating infected body cells, either in a helper T cell manner by coordinating the response of all of the other cells, or CD8 cytotoxic T cells actually targeting those infected body cells specifically, or cancerous body cells specifically, and eliminating those so that that infection can no longer persist in the body.